So it's taken us almost a year, but we finally got to what you guys thought biology was about, animals. That's right, we're going to start talking about lots of different types of animals, not just the cute cuddly ones, because it turns out there are a lot of just plain weird things out there. When you're on, I don't know, social media on Facebook and you start going down that rabbit hole of seeing the weird creatures, I don't know what the analytics give you guys to look at, but for some reason it always pops up to me. It's like, new deep sea creature discovered. I'm like, yes, we're looking at that for the next hour. And then everything related to it. There's a lot of interesting weird things out there. Before we get to those, though, let's talk about the characteristics of animals. Across the board, what do all animals have in common? And it is an amazingly long list. I'm sorry, Alyssa, what did you say? Okay, go ahead. Give us an example. That's right. Animals are multicellular, as you can see from the very first line of this slide. Well done, Alyssa. Animals are eukaryotic. They're heterotrophs. They have no cell walls. They have nervous tissue. They move. They reproduce sexually. They have an extracellular matrix. We'll get to that in a second. Tox genes, and very, very, very similar ribosomal. So even the most simple animal has many, many different cell types. What is that animal? It is Daphnia magna. Is it male or female? How can you tell? Well done, you win. It is female. It's got all those eggs in it. They're heterotrophs. They obtain their food by eating other organisms or the waste of other organisms. They take in food from the environment. This is a crab. It has mauled the turtle. That's right. Baby turtles get eaten. They get eaten a lot. Baby turtles don't have much life expectancy. That's why turtles lay that many eggs, because not many of them will survive it. On the way back to the sea, they have to contend not only with crabs, but with birds, with, once they get into the water, fish. Turtles have a hard life. Anyway, so here's a turtle being eaten. No problem. Animals have no cell walls. They don't produce cellulose. They don't have any lignin. So they cannot produce a cell wall, which means osmosis is an issue with animals. Even if you see something that has a hard carapace, like this lobster, it's usually made of keratin. Keratin is basically the same thing that your nails are made of. So that's not a cell wall there. What you're looking at is just an external coating of keratin. It fills the same purpose, though. Yes? Nope, we have no cell walls. We are not plants. Animals have nervous tissue. Even the simplest animals have a very, very simple nervous system, a way of sending signals from one part of your body to another very, very quickly. That lets you react to your environment. It lets you react to external stimuli. Like, Alyssa, hit Tom real quick. All right, see, he responded because he felt that. There was a sensation, he responded to it, he sort of moved away from the hit, or he tried to, and then he dropped his glasses on the floor and everything was terrible. Animals move. They have muscles and a nervous system which lets them at one stage of their life or another move around and do something. So this is, in case you couldn't know, this is a gorilla with its skin removed. Why? So you can see the muscles. You can't see muscles if the skin's still there. We hope. Yeah, he died first. Hey, did you guys know? Fun story. You know, you've heard the expression, there's more than one way to skin a cat? There's only one good way, just so you know. You start right here, you do a cut, and then you start peeling. Animals reproduce sexually. That means they have a haploid system where they produce gametes, and those two gametes come together to form a diploid zygote. They have an extracellular matrix. Extra means outside. So outside the cell, there's a lot of stuff going on. There are all these proteins out there that help bind cells together, but also allow cells to communicate back and forth. There's a lot going on outside of the cell, just like there's stuff going on inside of the cell. It's sort of like if you imagine two houses separated. They're two totally separate houses. What goes on inside the house is like what's happening inside the cell. But between the houses, they might share a lawn. Now, that lawn still has things happening, and both the houses, both the cells, are responsible for some aspects of that lawn. They have Hox genes. We talked a little bit about Hox genes when we talked about variation in the Cambrian explosion. These are going to be responsible for creating a body axis and body patterns. You'll notice that you have a left side and a right side. Awesome. Starfish can be bisected in many different ways. You have arms and legs and a head, cephalization. These are controlled by the Hox genes. Other organisms lack these. They have similar ribosomal RNA. The small ribosomal subunit of RNA is really, really well conserved. That means you and squid and rotifers, very, very simple organisms, all have basically the same ribosomes. Now, I put a picture of a squid up because according to the fossil record, multicellular life began around the Cambrian era. That's when we actually start seeing it. That's when we have fossilized evidence. Now, as you guys know, the fossil record is biased. If there were some really, really soft-bodied organisms around, they're not likely to fossilize very well. But we definitely have evidence about 590 million years ago, we have life, multicellular life. Now remember, the Earth is 4.5-ish billion years old. So for 4 billion years, not much going on as far as life goes. And then suddenly, 600 million years ago, we have multicellular life going nuts. Life may have proliferated, multicellular life really took off because of high ultraviolet radiation. That high ultraviolet radiation encourages mutation. So by having more mutations, you have more elements for natural selection to act on. During this time, the Hox genes developed. So those Hox genes, those body plan genes, allowed for more variation in morphology. Early in Earth's history, when we first had multicellular life, we didn't just have bilateral symmetry like we have now, a left side and a right side. There was all sorts of crazy pentalateral symmetry, hexalateral symmetry. You could be cut in many, many, many different ways, and you would end up having these mirror images. Organisms with 5 eyes, 7 eyes, 13 eyes, things like that. Question?
So, you know how we have layers in the rock?
Rock layers.
Yeah. So, if you weren't constantly digging and using the rock, would it be possible for the Earth to get bigger?
Good question. Okay, so his thought was, is the Earth growing? Can the Earth grow? And the answer is no. It can't continue to grow. Because what's happening is the way you get those layers is weathering effects. So you have different sizes of different elevations, different sized geologic structures. So they get weathered down and then create those layers. Now eventually, those are going to get subducted down. So now, quick geology lesson, right? Those get subducted down. So if you have 2 plates, one's going to go under the other. And as that one goes under the other, it's going to become liquid again. And then eventually, it cycles back up.
So what you're saying is it's possible that you have a whole nother era that you don't know about.
Oh, absolutely. It's possible that you have a whole other era you wouldn't know about. But the thing is, we have rocks that date back to the beginning of the Earth. I mean, so some things don't get subducted. You know, not everything gets pushed back under. So it's possible. I mean, unlikely, but possible. Then we got the Red Queen theory. Now, this idea was, the Red Queen theory is, again, Alice in Wonderland. You have to run as fast as you can to stay in the same spot. So in this case, animals evolved to fill a specific niche and compete with others. So as one of them developed, I don't know, as one of them developed a carapace, hard exoskeleton, another one developed the ability to break that exoskeleton, which caused the ones with the exoskeletons to maybe adapt to move faster or something like that. So there was one adaptation in prey led to an adaptation in predators, and so on, back and forth. Now, the very, very first animals are thought to have evolved from a eukaryote that resembled a choanoflagellate. Now, we talked about choanoflagellates at the end of the last lecture, so long ago. Choanoflagellates have this flagella this ring about them, this sort of collagen ring and sort of a head shape. Those choanoflagellates can come together to form pseudo-multicellular structures where they're all sort of hanging out together. They have cellular communication genes. They have adhesion genes, which are going to be really important for multicellularity, in case you forgot. Take a look at, so here we have sponges. Sponges live in the ocean. That always kills me. It's like, imagine how much higher the water level would be if there were no sponges. But, so if you look at the cells of the sponge, they're called choanocyte cells up here. Collagen ring, flagella coming out, and they're attached to another cell called an amoebocyte. But it looks like the same basic structure as the choanoflagellate. Yes? No. So the kitchen sponge versus an artificial sponge. So here's the story of the sponge and the loofah, because it all comes together. I'll tell you what, can you give me about 3 minutes? I actually think I'm going to get to that story, and it'll make more sense on a future slide. But I want to tell you the story. I really want to tell you the story. So the look is very similar. Now, that could be an example of convergent evolution. It can also be an example of direct evolution. They have almost exactly similar DNA sequences. So it's not just structures. They don't just look alike. They also have DNA similarities. Now, animals are typically going to be classified in taxonomy, right, by their body plan. So we're going to look at them by their tissues. Do they have tissue? Do they not have tissue? By their symmetry. Can you cut them in half? Well, you can't cut them in half, and they live for the most part. But can you look at them and they have a right side and a left side? Or do they have multiple sides? By the way they develop. And then two sort of ways we used to classify them that are out of favor right now are segmentation and whether or not they have a coelom. They could be classified by tissues. Tissues, like we talked about with plants, are just specialized groups of cells of the same type. So if you have heart cells together, you have heart tissue. Skin cells together make skin tissue. Does that make sense? So parazoans, para means almost but not quite, they have no specialized tissues or organs. They have no cells that are specialized to do things. Their cells are just distributed. Think the choanocyte and the amoebocyte, they hang out together. That's versus us, which are Eumetazoans. No, that's not us. But versus our group, which is the Eumetazoans. They have specialized tissues. They have specialized organs. Specialized regions of their body where things happen. So parazoans lack that. Eumetazoans have it. Again, para, almost but not quite zoan life. Eumetazoans, good middle life. Next, you can classify them by symmetry. Once you've divided them up into no tissues, parazoans, yes tissues, eumetazoans, you take those eumetazoans and say, what type of symmetry do they have? Now, they could have radial symmetry, like a hydra. You can cut it along many different planes, and the left side will look like the right side. In fact, if you cut this hydra up into 6 quadrants, each piece of it is exactly the same as the other pieces. Yeah, we, though, have what's termed bilateral symmetry. If you cut us straight down the center, our left half is a mirror of our right half. Does that make sense? Now, you can't do that on every axis. Obviously, if you cut us in the center, the top half doesn't look like the bottom half, except for really, really ugly people. And then all of you thought it. So you could have radial symmetry, group Radiata, jellyfish, hydra, what are those things called? Starfish. Bilateral symmetry, Bilateria, where it's a left side and a right side. Whether you have bilateral symmetry or you have radial symmetry, it's all determined by development. Now, back to the birds and the bees. After an egg is fertilized, sperm comes along, fertilizes the egg. That zygote will undergo cleavage. Lots of mitotic division. So if you have one and it splits in half, how many do you have? Two. Bilateral, thanks. Two. And if you have two and they split in half, how many do you have? And then how many? And then how many? 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 Good. One more. Good job. That's right. So split and split and split, and you very, very quickly go from a single cell up to more than 1,000 cells, and that happens in just a few hours. So as you're cleaving, those cleavage events are eventually going to lead to a hollow sphere called a blastula. So think almost like a balloon. You start off with a balloon, and it's this small point, right? As it expands, it goes outward, but the center remains hollow. All the cells are attached on their edges, not in the center. They're not a ball. They're a hollow sphere. So that hollow sphere is called a blastula. This is called blastulation. It's just a fun Scrabble word. It's only a fun Scrabble word because if you can get blastula out and then you add shun, T-I-O-N, you totally win. Blastulas lead to gastrulas. So the way this works is you have that hollow ball, that blastula, and then that blastula invaginates. It's almost like taking a balloon and sticking your fist into it. When you do that, you've got a curvature. So you have this hollow sphere that now has something going into it, which you see here with this gastrula. So you have an outside and an inside. That's going to be super important for animals. You don't want to be just a hollow ball. You want to have this hole. And that hole eventually is going to act as both a mouth and an anus. Not in you guys. Your mouth and your anus are different. That's an adaptation that we came up with later, and that's, as you can imagine, super important for you. This forms a primitive digestive system. Look, the deal is, though, as you're developing, you didn't start off with a mouth. Oh, no. You had an anus to begin with. So that's a gastrula, and that process is called gastrulation. So you're going to go zygote leads to blastula, blastula leads to gastrula. Gastrula starts to lead to your different cell layers, which is a great segue into this slide. There are 3 cell layers: the endoderm, the mesoderm, and the ectoderm. They're going to be defined by where they're located along this organism, or this developing organism. If you can see the blastula undergoing gastrulation, and you've got a distinct inside here, and outside there, and then a bunch of middle stuff growing in the inside. The outside layer is called the ectoderm. The inner layer is called the endoderm. And that middle part is called the mesoderm. So ecto out, endo in, meso middle. The endoderm is going to end up forming a very, very primitive digestive tract. The endoderm, all of those middle cells, they form the lining of the digestive system. The mesoderm goes on to form your muscles and your tissues and all of your internal organs, anything that lies between that digestive system and the rest of your body. Because really, think about your digestive system. In case you couldn't tell, this is a frog. Think about your digestive system. It's just a... I know everyone's like, the internet's a series of tubes. You are literally a series of tubes. You've got this tube that goes from your mouth down to your stomach, from your stomach to your small intestine to your large intestine, and then out the other end. Literally, it's one big long tube. Now, between that tube and your skin, you have other things. Lungs, a heart, appendix, kidneys. Excuse me? Blood tubes like veins and stuff? Yeah, pretty much. Those are another series of tubes. We're going to get to that when we talk about circulatory system. But the idea is the mesoderm is anything between the digestive system and the outside of your body. The ectoderm becomes your epidermis, the outer covering, your skin, and also your nervous system. So your nerves, your brain, things like that. Okay. So there we go. That's basic animal development. Unfortunately, this whole group called Radiata, they don't have 3-cell layers. They're not cool like us. Oh no. They only have an ectoderm and an endoderm. They're missing the mesoderm, which means what else are they missing? Right, they're missing muscles. They're missing tissues, organs. They don't do terribly much. They have a cool nervous system, though. Radiata are going to be like your jellyfish. You love the jellyfish? Oh, we'll talk about jellyfish soon. Another way to scientists have historically classified these is by the presence of segmentation and a coelom. The coelom is a fluid-filled sac, but turns out a lot of organisms have this, some don't, and it's an example of convergent evolution. So we can't actually, because of molecular data, we can't divide things up like this anymore.